going to move into some of the work I started presenting last year, and then if we have time, I'll also talk a little bit about genome instability in the germline, both in pre-implantation embryos and in eggs. But the first thing I want to go, and I think just really update you on, is the work we've been doing to try and understand the J-curve of aneuploidies. So I don't think I need to show this slide here again, but I feel a little bit. But I just want to emphasize at this stage here that when we're looking at eggs from women who are coming from fertility clinics, we don't really get them very young. And we really wanted to go and get the early J as well, because there's certain predictions that are coming out of the models that we are thinking of, particularly cohesion loss and other things. That's very, very clear that the mechanisms that are driving aneuploidy rates should be different. And in particular, the recent work from Francesca Cole from juvenile male mice that suggests that aneuploidy rates are very, very high in the first couple of meioses. And because of the covariance of female age and male age, she suggested in a paper that perhaps this bleep here is actually coming from males in the human population. So we've wanted to examine what drives the J and whether it is a J. And we've been using and setting up and validating DNA single-cell sequencing using chorial cell lines that had 13, 18, and 21 trisomies to make sure that what we're getting is right. So we take all sites of polar bodies, and later on I'll also show you some embryo work. We take single cells, the polar bodies, and I think the big critical step here is that we're all debating is whole genome amplification. We then make our libraries, and we can then either by shallow next-gen sequencing or by SNP arrays, we can then go in theory, we can get all of the aneuploidies, but we can get everything down in theory to single cell, to variants, to SNP variants. And the model that the meiosis field, I think, have kind of synthesized is this two-step model that Terry discussed. And the first one is that recombination that tethers homologous chromosomes together occurs during fetal development. The other thing that happens there as well is that S phase establishes cohesion, and together these crossover recombination events together with the cohesion between the sister chromatids form these bivalent structures that we can see under the microscopes when chromosomes compact in metaphase. So I've just kind of illustrated here. It's actually diffuse at this stage. But these are the structures that have to be maintained until puberty and the ovulation when meiosis 1 occurs. So we're talking about an arrest that's lasting 13 to 51 years in human females. And the second meiotic division then occurs upon fertilization when the sister kinetochores segregate. And the thought is that if you, for example, have a single crossover and if you have cohesion loss over time, along the arm in this case, you'll end up with bivalence, the bivalent deteriorating into univalence. And with respect to the chromosome machinery, or the cell machinery that needs to then separate and segregate the chromosomes, you can see here you end up where you just started. And we know there's a maternal age effect on seeing univalence and a low cohesion environment in general in human. So this is the first study came out from Ross Angel. This is a very small data set on M1 oocytes. And then also more recently from Melina's lab, where they showed a slightly different centromeric cohesion loss or low cohesion environment in oocytes that are older. And then the work that we've been doing together with Melina as well. So the thought is that, of course, that if you now have high recombination rates, it should be protective and against aneuploidy, particularly in women of advanced maternal age, generally as well, perhaps, particularly, of course, we don't know what the cohesion loss is over time, whether it's the same in everyone, in every egg. But I think Stephanie showed that when you look in the Down syndrome population and you compare recombination rates in a Down syndrome person compared to their siblings, you see that they have lower recombination rates genome-wide. And we showed directly in eggs together with Alan that indeed eggs that have higher recombination rates have better chromosome segregation outcomes as well in women of advanced maternal age. So I think that the evidence for this two-step or two-head model is very much, I think, one that's building, and I think it's worthwhile considering and testing. So we've been wanting to look at this shape for quite a while, of course, and we really wanted to get down here. And the way we did it was we decided to use a two-pronged approach. So normally what we do is we get these eggs from large and mature follicles from women who've been stimulated with exogenous FSH or other gonadotrophins. You get GVX, you can also get M1X, and you can also get eggs that are at M2. However, we wanted to look at eggs from the same woman, and a lot of them, because we know that recombination rates, they vary by as much as tenfold in oocytes, even from the same female ovary that we get out. And at the same time as well, I think it follows, or I think as a correlate or covariate, we know that recombination rates co-vary with axis establishment. So that means that there must also be a tenfold difference in how cohesin is loaded, given that recombination rates and length of chromosomes co-vary. So we were really interested in trying to see if we could get into these small antral follicles as well and take the eggs at a very, very early stage of follicle development. And if we could use this to kind of look at meiosis, particularly in younger females as well. So the way we did this is I moved my lab to Copenhagen 2.5 years ago, and one of the major reasons for that was because of Klaus Jüding Andersen, who runs our ovarian cortex preservation project or freezing project for girls and women who have blood disorders and cancers. And we get ovarian tissue, and then they freeze the cortex where the primordial follicles sit, and then they can be autotransplanted later on when they're in remission. So we've done over 1,000 in Denmark the last 50 years, not we, Klaus has, and over 100 women have undergone this, and 99% of them, they get their HPO axis back. There's also been babies born after this procedure as well. So it means, though, that because they can't freeze the medulla, we get all of these small antral follicles that are between 0.5 and 3 millimeters that are released. And we particularly get them when they're part as these cumulus complexes, cumulus oocyte complexes. So when I reported last year, I think we were quite preliminarily testing that the aneuploidy rates were similar or not too dissimilar from what we see from the IVF population. And in total, over the last 2.5 years, we've now collected 3,000 oocytes or complexes in order to look at their meiotic potential to undergo the meiotic divisions, and therefore also to look at chromosome segregation events. And you can see here that we collect from 3 different clinics, one in Warwick, one in Invicta in Poland, and in Genera in Italy. This is the Copenhagen clinic where we get the ovarian tissue, and you can see we're now getting younger individuals coming in here as well. We don't get that many eggs from them. I think that's what one would expect. And I have to say, below the age of 12, we haven't really gotten the eggs to undergo meiosis. It seems that with the in vitro maturation program we have, they don't respond as well as those from women or females who are older.
Sorry, Eva, you do get immature eggs from the 12-year-old?
Yes, we do get immature. So these are immature eggs, and then in the cumulus oocyte complexes, but they don't really do a lot. So this is the outcomes. I don't want to break all of this down. There's a lot of data. I just want to show you here that on average, when we take everything we get out, even things that people normally don't, but we've taken all oocytes, we get a maturation rate for the women or females who are above 15 years, we get them at about 30% that undergo meiosis 1 and kind of get you these mature eggs that we can then go in and analyze. So we then use the single-cell DNA sequencing pipeline that we've set up for research genetics. We're not saying anything about clinical use here. We're just using it to look at meiosis. And as Nathan showed you, we take both the polar body one and the egg. And the reason we do that is because we like to see these mirror a reciprocal gain and losses. So here for chromosome 1, you can see a gain and you can see a reciprocal loss. And basically everything that we are seeing that are whole chromosome-based are reciprocal, and they're coming therefore from meiosis. So I just want to share with you how we've then gone about this. So we've divided the curve into 3 groups. The under 20 females, so they go down there at the moment 12 to 17. We have 5 females from there. Then there's 20 to 32s and the 33 to 41. So I'm going to refer to them as young, mid, and amas. You can see here the proportion of oocytes, they are now higher, and we have been bootstrapping here. We only have 8 that completed meiosis 1 that we got really high quality data from. And that's one of the things we do want to firm up. But you can see here as well now that we are starting to see a U or J. And you can see that as well when we treat chromosomes as individual entities, the data we have here as well. You can see it's higher now in the young compared to the mids.
What is higher?
The aneuploidy rates.
Okay, so aneuploidy.
Sorry, that's, aneuploidy, rates.
The young women are having more aneuploidy.
Yes, younger women per chromosomes.
Those 2 plots. Okay.
Yeah.
That's higher than the advanced maternal age.
Yeah, at this stage, yeah. So the maternal, I would say the advanced maternal age group, when you go in and look at them in IVF clinics, something that hasn't really been discussed here is that in the chapter that Alan and I co-authored, it's very clear that when you look at the curves, the age curves in IVF population, it's shifted compared to what you see for the natural conception. And I think we still have to try and understand why that is.
What do you mean it's shifted?
It's both elevated in amplitude, presumably because we can now detect both monosomies that are not coming through in natural, in clinically recognized pregnancies, but it's also shifted right. So the onset of the acceleration exponential part of the curve seems to be shifted later in the IVF population. So it's one of the things that, that we're trying to figure out whether that's just a composite because we're looking at different chromosomes, all of them now with different curves, as was discussed, right? Whereas in natural conceptions, we're looking at a handful of them, but I think it's there are many explanations for it. I don't think it's stimulation, but I'll show you that later. I think it's genuinely because we're seeing different curves to different chromosomes.
In vitro maturation, do you think it might be more sensitive to age than in vivo?
No. No. I'll show you a slide later. If there's an in vitro. So I should as well say that the in vitro maturation we're using at this stage is different from what happens clinically when you get GVX from fertility from IVF labs. But I'm going to show you data where I compare the populations later. Yeah. I also think that Maria Scribe from the EV clinic has done some really wonderful work on the GVs. The GVX that you get from larger mature follicles from IVF patients. So we're talking about 15 to 22 millimeters, right? She's shown that if you take the ones that are fast maturing, that means that they have GV breakdown within 4 to 6 hours, and then they do meiosis 1 or metaphase 1 and complete it, everything completed there within 24 hours, the fast maturing fraction. Then you get all of the same outcomes as you do with M2s that were matured in vivo with regards to aneuploidy, pregnancies, and live birth.
Just one point
Yeah.
before you go on. That left-hand curve people keep reproducing is simply wrong. It may have been found in your study, in that particular study, but nobody else has found it. If you take all of the literature together on birth prevalence of trisomy 21, it is certainly not a J. And they found the same in the big British registry. It's just simply not a J.
Yeah, those are live birth, right?
Yes.
Yes.
No, not just
No,
like births.
prenatals as well.
Prenatals as well.
Okay,
We don't see
but
J.
that's for what, Down syndrome only?
Well, we only know.
Right, because they clinically recognized that. I mean, I don't know if Terry's here, but but he can answer to that. I mean, it's a composite, right, for other chromosomes too, that then breaks down further.
That curve on the left is a composite.
Yes, yes. So this is what I think this is also a really important point, right? When we start making composite curves, right, the curve changes shapes.
Yeah.
Yeah.
right?
Yeah.
And so,
Yeah.
So that is the proportion of chromosomes. No,
We've just multiplied the number of oocytes, yeah, to get the per chromosome rate,
chromosomes,
making mistakes in the eggs from the younger, yeah, on average, yeah.
not just that there's more oocytes in it, but there's actually more.
Yeah, but we only have 8 oocytes in one, but they are from 5 different females, right? So we're trying to increase that data set. That's really the rate limiting one to get because, you know, they're very, very rare. So what I think we have here is that certainly that we have an elevation in the eggs from the young females on a per chromosome basis too, and therefore I think we now have a model system that we can go in and actually start really querying the two-step model with regards to the J shape of the human aneuploidy curve. So what we started noticing when we broke the errors down that we are seeing is that we are now seeing 2 types of error rates that are occurring. So the first thing that we're seeing is that precocious separation of sister chromatids, where you can see here that the 2 yellow here have divided precociously. They shouldn't. You can see that increases with age, whereas meiosis 1 non-disjunction is quite high initially. Even when we bootstrap and we take an egg out that had a lot of errors, we're still higher than the mid to the AMA groups.
Can you give a percentage for those?
Sorry?
A percentage for
Yeah, it's 0.6 or 6% of the chromosomes are making a meiosis 1 non-disjunction error. Yeah, that's
from 8
right.
eggs.
Yes, from 8 eggs, yeah. There's 1 egg that made a lot of mistakes, but even when we take that out, we're up here about 4%, yeah. Yeah, so it seems to be that we have 2 curves. We have PSSCs are coming up, and non-disjunction is starting up high, and then it kind of comes down. And so we have, I think we conclude that we have 2 age-dependent error rates. That's giving us a very interesting shape. And I've just averaged for these are numbers for all chromosomes. I haven't tried to split them up at this stage. Okay, so how would that fit with what we see from the IVF clinic? So in the IVF clinic, we can't get the really young females below the age of 20. It's very hard to get them. But you can see here PSSC rates are higher than non-disjunction rates, both the mid and AMAs. And that's certainly consistent with that, what we're seeing in these small antral follicles as well.
So can I just ask you, what's the age of the, because you said it's 5 women, the youngest?
Yeah, they're 12, they're 12 to 17.
Right.
Yeah. Yeah. So, and again, I just want to emphasize we have 2 different maturation protocols going for the small antral follicles and for the large antral follicles.
And the young women were there for fertility preservation.
In the LAFs or SAFs?
Why do you have the tissue? Did they have cancer?
They have a whole range of things. The young females, yes, they have. But we don't. When we split them up by different. Breast cancer is the biggest diagnosis, so we rule out BRCA1, BRCA2 when we have them, because we know it's associated with POI. But we don't see, when we stratify by diagnosis, there are 4 different main diagnoses. We don't see any effect of diagnosis in this. And females who've had chemo before are not part of this data set either. Yeah. I think also because the women are getting their fertility back afterwards, after the autotransplantation, I think it's a pretty good indication of that their tissues are actually working okay, at least with respect to getting the HPO axis going, right? And the patients that we have here, there's 145. There's no indication. I mean, for the very young females, it's hard to say, obviously, but for the ones in the mid and AMAs, there's no indication of that there's anything wrong with their HPO axis coming in.
Data on miscarriage rates in teenage women?
So so we're looking at that at the moment for the miscarriage rate, and also recurrent miscarriages at the moment that are common. Reported, yes, exactly, yes. Yes, there's definitely the reporting. I mean, there's a social issue here, right? With the reporting.
That Danish study, wasn't it? No. enormous study of miscarriages. You know what I mean? And that doesn't see any, there's no special...
Yeah.
No.
Yeah, so Henriette Svar, who's the head of the National Recurrent Pregnancy Loss Unit and consultant at the National Hospital and at the clinic, she said that she thinks that there might be reporting issues, particularly for the very young females, the teenagers, who might not tell anyone that they're pregnant or might not even know they're pregnant. Right, and so I think the reporting of what they see clinically, right, is definitely here affected by that. Yeah.
Eva, you're going to talk about possible explanations or biological.
Yes. Yes, it's coming. So within the two-step model for age-related changes in the error types and what we think might be shaping the curve here, I think it's what we clearly see in the Down syndrome project, and also other types of errors that are coming out in the clinically recognized POCs, is one of vulnerable crossover configurations. And they're established during fetal development. So we think that's the first step, very much in line with the current model. So how we explain the two types of errors is that these chromosomes have a very high likelihood of making mistakes, right, when they segregate. But in the younger women where cohesion is relatively intact, they would do a meiosis I non-disjunction. That's the kind of errors they would make. In contrast, as women age, you end up with a situation where you see, where we see an elevation in a low centromeric cohesion environment. That's also correlated with this kind of bioorientation of kinetochores that we then propose is what precedes the PSSCs. And that would obviously fit very nicely with an age dependence in cohesion loss. So we wanted to test this because it very much predicts that the distances between these two sister centromeres as a readout for cohesion loss or low cohesion should be linear with age, female age, including in teenagers, if it really precedes PSSCs. So we have really nice data from mouse, from Mary's work with a long-lived strain, that that's the case. And we wanted to see whether this is also the case in human females.
Doesn't what Lena just presented also support why you see more PSSC? Because if with depletion of cohesion, your kinetochores now are more susceptible, which would give you PSSC.
Yes. Yes.
So
yes. There's nothing inconsistent so far. There is something that is, but I will get to that. Right, that we are testing, but not with respect to cohesion loss. But so what we did was we took M2X and we spread out the chromosomes, because you can really here see the distances very nicely at M2. And the prediction is now, do we see kind of a more linear, or do we see a U that follows the overall rates, right? But we would predict this should follow the PSSC rate, so it should be lower in the females who are below 20. So in this here, then in the midst it should go up. And it should continue increasing in the AMAs. So we've looked at 54X from different women, and you can see here what we're seeing now is that we see something that looks very much like it could precede PSSC. We certainly don't see a big smiley U for this at this stage here. So I would say that this is very much, I think, consistent with the idea that a low centromere cohesion environment is something that increases with maternal age, would be consistent with loss, and it's something that follows the PSSC curve. Okay, so I think this two-step model is something that I think really is happening in human X. There are a couple of unknowns still that we need to test, and the first one, of course, is these vulnerable crossover configurations coming through, in particular E0s. E0s are very hard to face. So unless there's direct facing, we don't really know when we get someone who's heterozygous for two maternal chromosomes whether they truly were E0s or whether it was a crossover somewhere. And we know that in adult oocytes, E0s or inferred E0s or potential E0s, as Alan and I call it in our paper, we can't face them either. we just see heterozygosity the whole way through. So it's one of the things that really we have to look at. We have to demonstrate, or we have to test whether we see E0s. There was one E0 reported from Sunny C's lab in China in their data on human oocytes, where they had sequenced the oocyte PB1 and PB2. But that's just one example. And I think it's one of the things that's a big prediction. We should see these vulnerable configurations. The other thing that we haven't quite come to grips with, which is really a little bit unclear, is that given that we see higher rates of aneuploidy amongst oocytes, even with the low data set, is that we can't really explain with a model, the two-step model, why we would see a decrease in the mids, right? So the question there really is, why might we see a decline in the mid? And there are two things that we are currently, pursuing. The first one is that perhaps there's something we're not seeing, another segregation error that also increases with age, like PSSC, but we can't detect it with our sequencing methods. And that's, I think, we don't have to invent anything here. Alan and I described it. There is a segregation error called reverse segregation, where both homologous chromosomes, they segregate their sister chromatids at meiosis 1, and we term this reverse segregation. It's occurring at 2 orders of magnitude higher than these, what one would predict from the single PSSC events. So they might have a different etiology altogether. But as you can see here, if they do that, it's invisible by the sequencing. We need to know that we now have green and yellow together as opposed to just gray. So we are genotyping a large set, and the current question is do we see for reverse segregation with respect to maternal age? So that's work in progress. The other, I think, issue that comes up as well is something that's been discussed a lot, and that's the idea of selection in the human ovary. And in particular, because we now have these small antral follicles, I just wanted to show you now that perhaps we can start looking at an in vitro system that leads to start answering questions about certainly certain specific hormones and how that might influence meiotic progression into the divisions, as well as the meiotic divisions themselves. So the data that I just want to take you through the data that we have here. It's a bit busy, but I have now aligned the aneuploidy rates per chromosomes that make a mistake from the small antral follicles compared to the large antral follicles and mature follicles that we see in IVF clinics for the young. So these are the below 20, for the mids, the 20 to 32-year-olds, and for the women of advanced maternal age, in this case 33 to 41. And we have an age-matched sample from M2 eggs that were matured in vivo. So these are the eggs that are being used for fertility treatment. They're coming from Genera and Invicta, so 2 different clinics that are reporting very similar data. And you can see here we're not seeing a massive difference between what we're doing with the GV in vitro, with in vitro maturation, and I think that's also what we would kind of expect, given Maria Scriba's data from the EV clinic. But what you can see now is that when you're looking at the small antral follicles compared to the large antral follicles, we definitely see an elevation both in PSSCs, but in particularly in meiosis 1 non-disjunction, the kind of errors that are coming through now. So at this stage, we can't distinguish between whether this is because they're coming from a different, different stage of follicular growth, and whether this is selection. But the other possibility, of course, is also that we're using different in vitro maturations for these kind of for the GVX.
Sorry, Eva, you're just saying in the advanced maternal age ones in the IVF clinic is just 20% abnormality?
2% of the chromosomes are abnormal. About 60% of the eggs have an abnormality. Yeah.
And
yeah.
2% of the chromosomes.
2% of chromosomes. Yeah. So I think the conclusions and where we are so far with the study is really that I think we're breaking the J curve into 2 error types and 2 curves. M1 non-disjunction, which has a high rate in young females and seems to decline with age, and PSCC, which increases with maternal age and appears to be correlated with the low centromere cohesion that I think that our models from the meiosis field would predict. We're also seeing, as I showed you in the last slide, that the large ones that may indicate that perhaps there is some selection going on with respect to how eggs and follicles progress in the ovary, but it's very difficult to say. It's within the limitation of having an in vitro system. But the data so far, at least, appear to be consistent with the 2-step model that we have in the field.
You get a variation in follicle size when you stimulate, right? But has anyone tried to correlate aneuploidy rates with follicle size?
I don't think with follicle size, right?
Because you can't follow it from...
I mean, when they go down to the smaller follicles, you get more GVs out, right? But with follicle size for them too, so I'm not sure. What they usually do is just report the follicle size in general, right? The average
The
range and average, yeah. So I don't think they have.
Just a thought. It'd be really lovely to be able to see what these oocytes from small follicles or from these younger women are doing. If we were able to look at what meiosis one looks like, or even live, to just see like what types of things are they doing to create these non-disjunctions, and does that actually look different than the mid to even older oocytes?
Yeah, we would very much like to do that too. And we can't send these eggs, unfortunately, we can't send them to Melina for the imaging. And as you know, the imaging on the human oocytes takes on a different... we've killed a few with various different imaging methods, and we just decided to stop.
Because you just kind of know the endpoint, but it'd be really cool to see like how they...
I completely agree. We would really like to see that too. And at the moment, we can't. It's also a little bit tricky because for the oocytes are coming out from the small antral follicles. The follicles are only 0.5 to 3 millimeters, and there are lots and lots of gap junctions. I mean, if you look at some of the data that some of the people have imaged, you can see there completely. So trying to get the cumulus cells off to actually try and do some imaging is very, very, very tricky. On them, so you know the maturation itself is quite necessary in this case, even though they are pretty much fully grown, certainly in terms of size compared to the GVs we get from the IVF clinics.
So the oocytes are, so this was sort of my next question, the oocytes themselves are similar, fully grown size?
Certainly just by size, yeah.
Do you, I think I've asked you this before, but do you think that there's anything different cytoplasmically?
Yeah, I mean, yeah. I do think that the cytoplasmic maturation will be very different in these eggs, right? I think one thing is to get the nuclei to divide, you know, and do a meiosis one. I think another thing is the cytoplasmic maturation that's important for embryo development. Yeah.
Eva, I still don't understand what's causing the high rate in young females. I mean, that's the observation.
Yes.
But
yeah.
why would we think, why would that be? I mean, is that the default? Because you start off young, okay?
So we don't actually know that the rates are indeed higher. It might be that the two-hit model can encompass everything, depending on what reverse segregation will do, right? So that's this lower one here, right? We don't know what they're looking like, and we're waiting for those data. So it might remain UE, or it might actually come up more like PSCC. We just don't know at this stage. And I think a very clean two-hit model would suggest that, would predict that the reverse segregation makes everything kind of, linear, and then, you know, but we just have to see what happens with, with the data. Otherwise, the higher rate, I'm not sure whether we have an explanation for it, but it could be anything to do with that. For example, the maturation of the eggs at that stage might not be as good. Certainly that's what we see when we go down to the kind of 15-year-olds and below. It starts getting a little bit more hit and miss what the proportion of eggs that actually make it through and mature. And as I said, below 10, I think we got 2 eggs that and they didn't pass QC on unfortunately when we sequenced. The polar bodies looked very, very bad as well.
Is there any variation from patient to patient?
Yes, there's quite substantial variation. Yes, also in how many eggs you get, etc. Yes, yeah.
So Eva, have you looked
Yeah.
at differences in cell death rate in the ovary between these age groups?
We haven't, no.
Okay.
We get the from the ovarian, from the cortex. We know how what that looked like.
But you don't get the cortex, you get the medulla.
We get the medulla, but from the cortex, when they prepare the cortex for freezing, they always do histology on a piece as well. So we have that to correlate with, and the ones that we've included all look really good. So, we haven't looked at things that didn't do good because it didn't look well. It's something to do, yes, absolutely, yeah.
And I presume you haven't looked at specific molecules or proteins related to this at all?
No, we haven't looked at trying to stain for specific molecules at this stage. Yeah, we haven't done that. No, we've been focusing on trying to get the rates out at this stage. Yeah, and the system, in vitro system, going. Yeah.
Is it possible with the, I think Terry mentioned it yesterday, was sort of the timing that maybe the older eggs are the ones that did recombination later, and the timing was off for them, I think you mentioned?
Yeah.
Is it possible in these early cohorts they did recombination, they exited that phase too quickly?
Yeah, so maybe. I'm going to sit on the fence. With the genotyping that we're doing, to see reverse segregation, we're using SNP arrays, so we're doing it genome-wide, and we'll be able to look at recombination rates, right, across a whole spectrum of ages now. So we will hopefully start getting an answer with respect to this ovulation timing and recombination connection, if there is one. Yeah, hopefully. Yeah. So I don't know if we have time, but I did just want to talk about the maternal to zygotic transition, which is characterized by high levels of genome instability. And that's certainly true in human IVF embryos. And we've been very inspired as well by some of the work Yorish has been doing. And Yorish came to review our department a year ago, and he kindly shared with us, or with me, that his in vivo data that he presented on the bovine, and that's now published, about that they had lower rates, right? So we were kind of interested in what happens and why the maternal to zygotic transition might be so unstable. And I know we're in an IVF system, but I think as we all recognize here, there's a big event that's happening. The zygotic or embryonic genome activation takes place after the first 2 or 3 mitoses. Everything else is coming from the egg. So we wanted to kind of have a readout for genome instability at this transition here, and we chose gross chromosomal rearrangements, as we call them in the DNA repair field. And these are large rearrangements. We're talking about greater than 5 megabases that we've validated on cell lines that we can pick up. And they are some of the syndromes here where whole chromosome arm is affected. So these are from about 800 or so trophectoderm biopsies, and these are just the uniform ones. I don't want to get into the mosaics about these, but you can see here that a fraction of euploid, and we have about 35% whole chromosome aneuploidies, segmentals. I've here just illustrated that an arm is missing, and the whole chromosomes aneuploidy segmentals. Their incidence is that what you would expect from 2 independent kind of events. You can see here as well that if we take the data and we now go maternal age, you can see euploid comes down, whole chromosome aneuploidies go up with the maternal age group, whereas the segmentals, they stay flat with maternal age. We also know that when we look at the chromosomes that are coming, we see 15, 16, 21, 22, for the whole chromosomes. You can see here for the segmentals, it's pretty much all of them coming, a few of the smaller ones notwithstanding. And that suggests to us that gross chromosome rearrangements and segmental aneuploidies have a very different origin than whole chromosome aneuploidies. I just want to show here the size distributions as well, and also want to say here that 27% of what we're looking at involves the whole chromosome arm. And we're going down to about 5 megabase. That's our lower resolution cutoff that we're okay with saying something about for research genetics. The gross chromosomal rearrangements that I've shown you here affected about 10% of the blastocyst trophectoderm. But I think if we model in now the data both from Vanessa and also Alan's recent data as well from the cleavage stage embryos that are rested, we end up with something like 16 to 24% of pre-implantation embryos that have in one of their cells a segmental aneuploidy and a gross chromosome rearrangement. And when Joris then came and talked to me and shared his unpublished data at the time, I got very nervous because of this. I wanted to think, okay, maybe this is in part due to in vitro, the way that embryos are grown in vitro. It could be, right? So what we decided to do was go back the whole way to eggs and see what we find in eggs, because we kind of thought, well, some of them might be coming from meiosis. And I don't want to go into all the sequencing we've done for this, but we've looked at both mature M2 oocytes, and about 5 to 7% of these have a segmental aneuploidy, and also in activated zygotes. There are 3 data sets out there, 2 from Alan, including the one Alan and I did together, as well as one where they made actual zygotes from Sonny C's lab. And there the rates are estimated to be about 18 to 13%. And the majority of these events are reciprocal between the polar body and the egg. So I think that we now have evidence for that these gross chromosomal rearrangements that we're looking at least some of them, are coming from our germline early on. So we wanted to see evidence for chromosome breaks, and I just show you here, this is a spread, and you can see here's one arm, here's another sister chromatids, and one of the arms are missing. And we see that at a rate that's not dissimilar from what we would expect from our sequencing data. We also collaborated with Agatha and Melina, who had data from their intact spindles, because when you start spreading chromosomes, although we have really great resolution, then there's always, some, are they really truly, did we introduce artifacts? So these are the spindles that you saw earlier from Lena, and you can see typically you see these very nice symmetrical structures, but we now Agatha and Melina start seeing these atypical structures as well in these eggs. And you can see here as well that the rate is very similar or consistent with what we see from sequencing. So we actually think we have gross chromosome rearrangements occurring and chromosome breaks in human eggs and potentially also the embryos later on. So,
can I ask something in the... So you see also the reciprocal segment. No, this is just the oocyte, or was it the polar bodies and the oocyte?
We couldn't, they can't image in the polar body, unfortunately.
Not the imaging, but also your sequencing, because you said...
Yeah.
So do you see the reciprocal?
Yes, we see it about, I think, 80% of the cases we see a reciprocal. Yeah, we do see, yeah. In my first slide when I showed the technology, that was a chromosome 9 segmental there that we used, yeah. I just want to go down to then say that what we're seeing in terms of gross chromosomal rearrangements and chromosome breaks is really what we see in transformed cancer cells and cancer cell lines that a number of people in our colleagues work with. And we've been wondering then where they might be coming from, these gross chromosome rearrangements, because it seems that the system that's being set up here in human oocytes are highly unstable. We wanted to look in mice, and in mice it's also quite high, the fraction of oocytes that have a chromosome break when we spread the chromosomes, but you can see human is higher than mouse in this case. And to start understanding what causes or what might drive genome instability, we started sequencing. So we've sequenced 101 human M2 oocytes, and I'm just going to show you here the data we have from women where we had multiple eggs, because this is where we can really see heterogeneity. So this, I've just here clustered the gross chromosome rearrangement genes. That's based on a lot of data from the DNA repair field. There's about 237 of them. There are transcripts per million here, and you can see here that they're eggs from the same donor. They're highly, highly variable from them. And obviously, we have heterogeneous expression now, and the big question is it functional? Does it matter? So we've been thinking for a long time how we test this system and how we test the importance of heterogeneity, particularly in an in vivo model. And we wanted to do it in vivo because we wanted to see that this can actually happen, not just when we treat things in vitro. And we decided to look at gene dosage systems, where we reduce a gene because we reasoned that this would then cause any imbalance to be amplified. And the system we chose to do this in was mouse, and we asked the question, do we now get rates that are higher or elevated, so they look more like human rates or even higher than that? And the system and the gene we targeted for the proof of concept was SMC56. So we know a lot about SMC56 in chromosome integrity from the yeast, both yeasts, as well as C. elegans and Drosophila, a number of model organisms. We know what it does very, very well. It's heterogeneously expressed in human X, and when we looked at the breeding data we had for a gene trap model at the MRC center where I used to be, I found that about 20%, we saw a 20% reduction in the litter sizes. So we looked at this gene, the eggs from the gene trap females, the heterozygous. We did a lot of characterization, and you can see here that now we start seeing these chromosome breaks in M2 oocytes that are now elevated in the heterozygous, in oocytes from the heterozygous mice. And they're often kind of centromeric or pericentromeric. Here you can see we are missing one, and here you can see that we have an extra. No. Yeah, these are just M2. Yeah. So I think with that, we kind of think that at least some of the heterogeneity in gene expression that we're seeing in human X might actually be causative of gross chromosome rearrangements in human X as well, on mammalian X. And we also have a molecular model in this case. The 237 genes do very different things. So this is why we have to be very, very specific. But we think that they're coming from the resolution or rupture of these anaphase bridges that we see in M1 oocytes in the head. We see it in wild type as well. We also see it but we see it with an elevation in the eggs from the females. I don't want to go into too many details at this stage, but we do know that any kind of unresolved bridges at this stage here, this is metaphase, but at anaphase as well, the resolution or the dissolution, etc., would give rise to gross chromosome rearrangements, depending on how it's resolved. And I should also say that there's a whole body of literature from the recombination field, that molecular recombination field, that we know that these chromatin bridges, generally speaking, that they cause gross chromosomal aberrations. So I think the model we have is that for SMC56, at least some of the heterogeneity, you get these bridges, they shouldn't be there. And then at the first division, you end up with a break, you get fertilization, and you'd have an eccentric chromosome at this stage here. So with that, I just want to say that there's, of course, also implications for embryonic mitosis now, given that the egg is going to drive the first 2 or 3 mitoses. It'll coincide as well with the embryonic genome activation. There's really high level of noise that's coming through in the gross chromosome rearrangement network. I would also like to say, of course, if you've only looked at really gross aberrations that are occurring, right, and that other kind of smaller things, duplications, deletions, we're not picking up because of the resolution of our sequencing. Gross chromosomal rearrangements.
A single break, it's just like a 20 megabase deletion.
Yes,
deletion,
yes.
Right? But it's not like you see all 5 chromosomes which are
No.
scattered.
No, we don't see chromathripsis. It's just like a single event, yes. Yeah.
But these numbers fit with what we see in Day2.
Okay.
The second cell division.
Yeah, okay.
Holds everything there.
Yeah, okay, yeah. And okay, so where would the noise come from, and might this also impact what happens in the human embryo? And I think that there's now data that are coming up from 3 different labs that have shown that mammalian oocytes are very, very special in the oocyte during the growth phase and into M2, they set up these really broad domains of H3K4 trimethylation. Now, this methylation mark is important and it's required for transcription, and it's set up all over the genome. So we think that 20% of the histones are actually methylated in this way. You can chip it, and you can see that this is our collaborator from Oslo, and you can see in sperm they don't have it. The 2-cell stage, you can see they start to be removed, these broad domains, and at the 8-cell stage and in human embryonic stem cells, they're very much confined to these transcriptional start sites where they're supposed to drive EGA. So one possibility, we think, is that perhaps setting up these very broad domain in the eggs might cause a lot of the transcriptional variants because of spurious transcription coming where it shouldn't come.
Then there's a problem because this actually go back for the segmentals, but we see both maternal and paternal equal amounts.
Yeah, yes, but the paternals could still come from chromosome breakage, right, in embryo.
Sure.
Yeah. I mean, I don't think this precludes it, and I'm not saying there's just one mechanism. There might be several mechanisms. Yeah. So with that, I just wanted to thank the participating clinics, and in particular also, of course, our collaborators, Alan and Melina, were supposed to be here as well. This is the lab. A lot of the sequencing and developing, adapting it for single cell was done by Bobby, who is the shared PhD student at Illumina between Alan and myself, our funders, and of course all the girls and women who've participated. Yeah, thanks.